dear friends. So our today's topic is base excision repair. So this is the type of repair mechanism of DNA in which any wrong base is excised and then the DNA repair is done. For example, let's see, suppose this is the DNA molecule. This is the one strand and this one is the another one. And here we have got a base guanine and it has been paired rightly with cytosine. Sometimes one base gets changed into the another base due to many mechanisms that we'll discuss later. In this example, let's say the cytosine has changed into uracil in the DNA molecule. Now our cell has to remove the wrong base, that is uracil now. So let's see how it's done. This is our sugar phosphate chain. This is sugar molecule phosphate, sugar phosphate and sugar phosphate. This is one nucleotide made up of phosphate, sugar and one nitrogenous base. This nitrogenous base has changed from cytosine to uracil in this present example. Now we'll remove this uracil. This sugar moiety and nitrogenous base, they are bonded to each other with the help of N-glycosidic linkages. See, this is the sugar phosphate backbone of the DNA molecule. Sugar phosphate, sugar phosphate, sugar phosphate, and the nitrogenous base is attached to the sugar moiety with the help of N-glycosidic linkages. And there is phosphodiester bond in between the sugar and the phosphates. So in this case, if we have to remove this wrong base, then first we will have to break this N-glycosidic linkages. So first of all, enzyme uracil DNA N-glycosylase comes in the present example. This enzyme removes the wrong base, that is the uracil in this case, and we are left with the plain sugar phosphate, sugar phosphate, sugar phosphate backbone. So this is how the wrong base gets removed. Now we don't have any base here. We only have got the sugar phosphate backbone, and here where the bases were right, here and here, these right nitrogenous bases are still there. Only the wrong base which was attached here, that has been removed with the particular type of enzyme, that is the uracil DNA N-glycosylase in the present example. Suppose there is some other base that has to be removed, then that particular N-glycosylase comes in that case. So now we have removed the uracil and there is no base attached here now. So in this way, now we have got this site as the A-pyrimidinic site in the present example, or we can get it as the A-purinic site, depending upon whether any purine or pyrimidine base has been removed. Suppose the wrong base was any purine, then we will say now this site is A-purinic site. Suppose this base was any pyrimidine, then we will say this site is A-pyrimidinic site. So this is how we get the AP site. AP is A-purine or A-pyrimidine. A means without purine or without pyrimidine. So this is how now we have removed this wrong base. So let's see what happens next. Now this site we have got, that is AP site we have got. Now what will be the next enzyme that will come? That will be AP endonuclease, that is A-pyrimidinic or A-purinic endonuclease. So what does an endonuclease do? And endonuclease always makes a nick inside the DNA strand. So this enzyme AP endonuclease now makes a nick here. That is in the area where the wrong base was attached. Now we'll see, now we are left with this sugar phosphate and sugar phosphate. All the sugars and the phosphates are there, but this bond has been broken. That is one side of the phosphodiester bond. This bond has been broken. Because we saw this bond is the phosphodiester bond. Sugar moiety is attached with the phosphate molecules with the help of phosphodiester bond. Now this bond has been broken and we have got a nick here now. So let's see what happens next. Now we have to remove this moiety, that is the leftover part of the wrong nucleotide, or the nucleotide where the wrong base was attached. The base we have already removed. Now we will remove this part also, that is this sugar as well as this phosphate, means the leftover part of the wrong nucleotide. So what will be the enzyme? The enzyme's name will be deoxyribose phosphate lyase, that is deoxyribose phosphate lyase. Lyase means now this enzyme will remove the sugar as well as the phosphate moiety. So now what do we have now? Now this base, wrong base has been removed from this site. Sugar moiety has been removed as well as the phosphate moiety has been removed. So the whole of the wrong nucleotide we have removed, and now enzyme DNA polymerase 1 will come. So what does N polymerase do? Polymerase always attach the nucleotides in the 5' to 3' direction. So this DNA polymerase 1 will attach the new nucleotide from 5' to 3' direction with the right base, and whatever nick will be left, that is in the place where this bond was broken, that nick will be ligated with the help of DNA ligase. So this is how we'll get to remove the wrong base and we'll get the DNA double strand with the right bases. So let's see the whole process again. This is our guanine base that has attached rightly with the cytosine. Now in our cells, this cytosine has somehow changed to uracil. So to remove this uracil, enzyme uracil DNA N-glycosylase comes and removes this wrong base, that is remove the uracil, so that we are left with the AP site, that is apurinic or apyrimidinic site. To act upon this site, enzyme AP endonuclease comes and makes a nick here, breaking the phosphodiester bond. After this moiety sugar and phosphate gets removed with the help of deoxyribose phosphate lyase, and so that the whole nucleotide gets removed, then the right nucleotide gets attached with the help of enzyme DNA polymerase 1, and the strands or the broken portion of the strand are ligated with the enzyme DNA ligase. So this is how base excision repair gets done in our cells.